Hi, this is the first in the series of lectures on RNA editing. And to underscore the importance of RNA editing and all the post-transcriptional processing that occurs after the RNA polymerase makes the RNA, I wanted to point out that every year there is an Oscar given out for Best Film Edit. And that the movie industry would not be able to exist or produce good movies without a proper editor. And the same goes for the cell. So RNA processing, post-transcriptional processing, RNA editing, there's lots of ways to talk about it. There are post-transcriptional modifications. There's lots of terms that are used to describe the three processes that occur with eukaryotic RNA after it has been transcribed, or as it is being transcribed, as you will see. It's the final step in the production of the mRNA molecule that is then will be translated into protein, and so is the part of the central dogma of molecular biology. Now, bacteria do not process their mRNA at all. That's a very big difference between eukaryotes and bacteria. And the reason they don't process their mRNA is because they don't have the problem of a nucleus or the problem of introns. So here's a typical RNA transcription process in bacteria. The RNA polymerase is running along the DNA template. It's making RNA. And as soon as the mRNA is being made, it's instantly translated by ribosomes and proteins are produced from it. As a result, bacteria can divide and can grow and divide much faster than eukaryotes do because their mRNA transcription, the transcription translation process in bacteria is much faster because there is no barrier. And you can see it here on this on this photograph by electron microscope. So there's a transcriptional site here, and you already see there's already a little bit of RNA has been made, and promptly there's two little ribosomes that are sitting on it translating the protein. So the DNA is running along here. That's the template. All the black things coming off of it is actually RNA. Somewhere underneath all those ribosomes, there's still RNA. You can kind of see it. But you can appreciate that the RNA is promptly coated with the big blobs of ribosomes that are translating the message. In eukaryotes, the steps are different. As I mentioned, eukaryotes have a nucleus as a result, and they have introns. Both of those things need to be accounted for. So the first thing that happens is when RNA begins to be processed is a 5' cap is added to the very beginning of the mRNA molecule. Then as the second step of RNA processing is to splice out all the introns and fuse the exons perfectly so they create proper frame for translation. And then finally, at the termination of the RNA transcription is also the last step in RNA processing. The transcription is terminated when the RNA polymerase encounters an polyadenylation signal sequence, the AUAA, whatever. And at that step, you saw that the polyadenylation machinery binds to the poly-A signaling sequence, chops off the rest of the RNA, and adds a poly-A tail to it, while the rest of the RNA is degraded by X nuclease that removes it out of the DNA polymerase and causes dissociation of the DNA polymerase from RNA polymerase from the DNA template. So RNA processing factors. So RNA processing, the three steps I just outlined, and RNA transcription are actually tightly linked with each other. And all of the three different types of processing factors, the first splicing initiation factors, the polyadenylation factors for the tail addition, and the capping factor for the 5' cap addition, are riding along on this on the RNA polymerase tail. In this figurine, they really did show it as a tail, as this really long, skinny thing. It looks like a mouse. It doesn't actually look that way. So instead, if you remember, this is a schematic of a eukaryotic RNA polymerase 2, and this is what they mean by the tail. It's the C-terminal domain, which consists of two proteins, two RNA polymerase proteins this C-terminal domain is phosphorylated when the RNA polymerase is allowed to clear the promoter and begin transcription. And the tail is also the place where all these processing factors are localized, so that they can process the RNA molecule as it comes out of the RNA polymerase machine. So let's look a little bit closer. This is sort of a mnemonic that I think is helpful to really remember, that mRNA is not good for translation unless it has a 5' cap and a tail. It can have some misspliced exons and introns inside. That can happen. That's a mistake of the splicing machinery. But the RNA processing, the capping and polyadenylation are critical to the RNA's existence. It will not exist. It will instantly degrade it unless it has a 5' cap, a protective 5' cap, and a protective poly A tail that the exonucleases found inside your nuclei don't degrade the RNA that was just produced. Sort of a mnemonic here is that RNA is a fashionable molecule. It has to be well dressed before it goes out into the cytoplasm, out in the town, so it has to have a top hat and tails in order to be seen and be translated. So there they are. There's the 5' cap. It protects because it's such a strange. It's a GTP edited in a strange direction. We'll see it in a second. And the poly A are protective modifications for the RNA molecules, preventing its degradation. So the 5' cap is added by a protein with a fantastic name. It's guanylyl transferase. And this whole thing is the cap. And it's basically, you can see it's a GTP. So there's a guanosine, there's a guanine here. This guanine happens to be methylated. The riboses of the first few bases of the RNA molecules also are sometimes methylated, but you guys don't need to remember that. So anyway, the guanine is methylated. You can see that this is not a deoxyribose, but a proper RNR ribose nucleotide. So it's GTP, not DGTP. And then you can see that all three phosphates here are intact, and a 5' to 5' linkage has been catalyzed here. So the 5' the 5' carbon here and the 5' carbon are what facing each other. That's why it's called a 5' to 5' linkage. The 5' cap is added almost instantly as soon as the RNA is synthesized by the RNA polymerase, because, as you just saw, the guanylyl transferase is sitting on the C-terminal domain of polymerase II. And the RNA, as it's being made, is passed by it. The cap the guanylyl transferase will bind the first nucleotide here. This is the first nucleotide, and then add the GTP, the inverted GTP, creating a cap. Then the guanylyl transferase will leave and dissociate, but the presence of this guanylyl cap, the 5' cap, will recruit the cap binding complex that will continue to actually hold the growing mRNA molecule in vivo in a nucleus to the RNA polymerase. Now, all the electron micrographs I saw you guys, that there was no CBC added to those mixtures. So the CBC was not holding there. There was no loops that you saw there. So that's basically the gist of the 5' cap. It's important to remember that it has this 5' to 5' linkage, but most important thing to remember that it exists, that it is important, that if you don't have a 5' cap, your mRNA is vulnerable to the exonucleases, RNA exonucleases that are present in the nucleus, because they're the ones that will be degrading all the introns and everything else, all the misprocessed messages. So 5' cap is critical for RNA survival and its ability to make it out of the nucleus. And here I wanted to mention something that you're probably going to talk in a much greater extent in biochemistry, but just the naming of the enzymes in molecular biology and biochemistry. You can see here that she's called a guanylyl transferase. So guanylyl describes the chemical group that it's working with. It's you saw it's a guanosine here. The second thing is that it transfers the guanylyl moiety to its substrate. So it has substrate plus guanylyl, and it will transfer the guanylyl onto the substrate. It doesn't modify the guanylyl; it just transfers it. The ACE is the usual way you would designate enzymes in molecular biology. If you see an ACE, most likely it's 100% likely it's an enzyme. So you guys can look at all of these different names that we have already encountered of the different enzymes found in molecular biology. The helicase is an enzyme because it hydrolyzes ATP. The ligase is an enzyme because it catalyzes the formation of a phosphodiester bond. And T5 exonuclease is an ACE because it breaks the phosphodiester bonds between nucleotides, and the exo denotes the fact that it does it from the outside. In comparison, so it has to attach itself to the outside, to the very end of a DNA or RNA molecule, and then start chopping from the end on. AP endonuclease is a different type of nuclease. It also breaks the phosphodiester bonds, but it can do so by recognizing something within a DNA molecule. So remember that AP endonucleases are the ones that find the abasic site during a base match, during the deamination repair. So it doesn't have to dock to the very end of a molecule of DNA and start chewing, but it can go in the middle. So endo means in the middle. And so forth. And I will give you an extra point on this quiz that we're going to have next week. If you send me three names of enzymes and describe the function